Hello everyone, welcome back to the class. In our previous class we discussed about role of p53 protein in cell cycle regulation. We started by discussing about which gene codes for p53 protein, that is TP53 gene that codes for p53 protein. It is a tumor-suppressive gene which codes for p53 protein. It is also called as TP53, which stimulates transcription of gene encoding a CKI protein, that is P21. Its activity stops the formation of tumor. It is located on human chromosome 17p13.1 and encodes a polypeptide chain of 393 amino acid residue. CKI protein binds to G1 bar SCDK and SCDK complex and inhibits their activities, thereby helping to block entry into the cell cycle. It hence, in response to DNA damage, p53 levels increases. It arrests the cell at G1 phase of the cell cycle. If all repairs are made to DNA, cell divides normally and completes the cell cycle. If cells still contain mutated or duplicated DNA sequence, it dies by a suicidal apoptotic mechanism to prevent its proliferation. In normal cell, p53 is highly unstable and present in very low concentration. p53 is inactivated by a negative regulator, that is MDM2, that is mouse double minute 2. MDM functions as E3 ubiquitin ligase, which inserts the ubiquitin chain to the p53, causing the proteasomal degradation of p53. In cells which contains mutated p53 or lost p53 genes, it causes arrest at G1 phase does not occur, and the cells that have mutated genomes proliferate and become cancerous. DNA damage or stress, when there is a DNA damage or stress, various pathway dissociates p53 from MDM, making it active and induces cell cycle arrest to allow either repair and survival or apoptosis to discard this damaged cell. Protein kinase ATM is activated in response to double-stranded breaks in DNA, which in turn activates CHK2 kinase. ATM and CHK2, this has to be CHK2, by mistake it is CHK1. Kindly apologize for this. ATM and CHK2 both phosphorylates p53 at distinct sites. Phosphorylation of p53 happens on amino acid residue in its N-terminal domain by kinase such as ATM and CHK2. So sorry for this mistake, it has to be CHK2 here. So CHK2, ATM and CHK2 alters p53 domain. This prevents association of MDM2 with P53. So this is the overall procedure how DNA damage arrests the cell cycle in the G1. There is a DNA damage, ATM or ATR kinase is activated, which in turn activates CHK1 and CHK2 kinase that results in phosphorylation of P53, allowing MDM to set free, and p53 will be active. Active p53 binds to regulatory region of p21 gene, and it results in transcription and translation of p21, which is a CDK inhibitor protein, which inactivates the G1 bar SCDK and SCDK. So this is the overall role of p53 protein in cell cycle regulation. So there is another simple topic, that is replicative senescence. So replicative senescence, will complete that topic and will get into cell cycle part 2, that is mechanics of cell division, in this class. So to begin with, there is a normal eukaryotic cell which have undergone several cell division. So is there any stop? Is there any signal to stop? Is there a limited capacity of that eukaryotic cell? Yes. So normal eukaryotic cell have only a limited capacity for cell division. After this limit, the eukaryotic cell will not undergo cell division. So the process that limits the cell division has been termed as replicative senescence. What it is? It is replicative senescence. So this replicative senescence limits, it is a process which limits cell division after a particular period of time. It appears to be a fundamental feature of somatic cell. So it is a fundamental feature of somatic cell with exceptions of most tumor cells and possibly certain stem cells. In 1960s, there is a person called Leonard Hayflick. He observed that human cells placed in tissue culture stop dividing after a limited number of division. So the limited number of division, or the number of mitosis a cell is capable of undergoing in tissue culture before it stops dividing, is termed as Hayflick limit. It is a limit of that particular cell which is undergoing cell division in the tissue culture before it stops dividing. So it is the number of mitosis a cell can be capable of undergoing in tissue culture before it stops dividing. So this limit of the cell is known as Hayflick limit, which was given by Leonard Hayflick. There is another term, that is telomere shortening. You have heard this telomere in structure of eukaryotic chromosome. So telomere is the terminal end sequence which is present in the chromosome, which caps the chromosome. So there is telomere shortening process. Why does this happen? What is its significance? So telomere shortening is considered as main causal mechanism of replicative senescence. The telomeres are short tandemly repeated. So these are short tandemly repeated DNA sequence that caps the ends of eukaryotic chromosome. So this serves as a dual role. What are those roles? First role is protecting chromosome ends. And the second role is intracellular signaling. So these are the two major roles. And what is the significance of intracellular signaling? It is for regulating cell proliferation. So the main function of telomeres is it is to protect the chromosomes ends, and in intracellular signaling for regulating cell proliferation. So a complex of 6 telomere associated proteins forms a complex. Okay, that complex is known as telosome. It is often called as shelterin complex. Now what is its role? So it plays a crucial role for both. It helps in maintenance of telomere structure, maintenance of telomere structure and its signaling function. And signaling function of who? Signaling function of whose? Telomere. So the function of telosome is also 2, that is maintenance of telomere structure and its signaling functions. And the length of, consider this is the length of telomere DNA. The length of telomere DNA is maintained by an enzyme, that is telomerase. It is a reverse transcriptase enzyme. It is a reverse transcriptase enzyme. It maintains the length of telomere. By overcoming end replication problem. Okay, most human somatic cells are telomerase negative and thereby experience this progressive telomere shortening. As because of telomerase negative condition, they experience progressive telomere shortening. Okay, if there was a telomerase enzyme in human somatic cells, so then there will be no question of telomere shortening. So as the telomerase enzyme maintains the length of telomeric DNA, if telomerase is present, then the length of telomeric DNA is maintained. If telomerase is absent, then there is progressive telomere shortening at each cell division due to the end replication problem. Why this telomerase shortening happens? Due to end replication problem. This end replication problem will be dealt in detail during DNA replication chapter. Okay, as for now, just remember telomere shortening is due to end replication problem. So as human cells proliferate in culture, their telomeres get progressively shorter and shortened down to the point when they elicit a DNA damage response. If there is a DNA damage in between, so then it will lead to deletion or progressive shortening down to a point. So what happens after this? So after there is a DNA damage, it activates p53 dependent cell cycle. We know that resembles the arrest caused by other type of DNA damage. So it leads to an irreversible growth arrest, a phenomenon which is called as cellular senescence. So now how to maintain this telomere? So telomerase therefore constitute a telomere maintenance mechanism conferring infinite replicative potential. However, in some cells telomerase is absent. In that case, who will take care of this mechanism, or who will take care or who will maintain the telomeric DNA? It is by a non-telomerase based mechanism. It is by non-telomerase based mechanism through which the maintenance of telomeric DNA is taking place. That mechanism is particularly called as alternative lengthening of telomeres. RES, okay, alternative lengthening of telomeres. In short, we will call it as ALT. It was first discovered in yeast, that is Saccharomyces cerevisiae. This ALT mechanism is also used by the minority of human tumor cells that lack significant telomerase activity. So the available data indicate that ALT involves in homologous recombination mediated DNA replication, which provides a solution for end replication problem, which provides a solution for protecting the ends, which provides a solution for progressive telomere shortening. So this was about replicative senescence. So now let's look into the notes part. Replicative senescence is a process that limits the cell division. It is a fundamental feature of somatic cells with the exception of most tumor cells and possibly certain stem cells. Leonard Hayflick observed human cells placed in tissue culture stop dividing after a limited number of cell division. So the number of mitosis cells is capable of undergoing in tissue culture before it stops dividing is known as Hayflick limit. The main causal mechanism for replicative senescence is telomere shortening. Telomere is a short tandemly repetitive DNA sequence that cap the ends of eukaryotic chromosomes, which protects the chromosome ends. It plays an important role in intracellular signaling for regulating cell proliferation, and also it maintains the telomeric structure, and also it maintains the protection of ends. Okay, complex of 6 telomere associated protein, which is known as telosome, or it is known as shelterin complex. It plays a crucial role on both maintaining the telomere structure and its signaling function. Telomerase is an enzyme which maintains the length of telomeric DNA. It is normally telomerase expressed in stem cells, tumor cells and germline cells, but absent in somatic cells. So if it is absent in somatic cells, so there will be another mechanism which helps to maintain the telomeric DNA, that is alternative lengthening of telomerase. So this telomerase is a enzyme which is reverse transcriptase enzyme which maintains the length of telomere by overcoming end replication problem. It activates p53 dependent cell cycle arrest. It leads to irreversible growth arrest called cellular senescence, constitutes a telomere maintenance mechanism, conferring infinite replicative potential. So this was about replicative senescence. So now we shall start with the part 2 of cell cycle, that is mechanics of cell division. Yes, in this PPT there is mechanics of cell division, and there is difference between mitosis and another cell division, that is meiosis. Stages of mitosis, that is prophase, metaphase, anaphase, telophase, and at last cytokinesis. So it is in detail we'll be learning in this class. Okay, to begin with, in eukaryotes there are two types of cell division: partition the genetic material into progeny or daughter cells. So there are two types of cell divisions. Two types of cell division, namely mitosis and meiosis. Mitosis is known as equational division. Equational division. Meiosis is known to be reductional division. Reductional division. Why this type of distinction between mitosis and meiosis is based on the fact of genetic material. Genetic material. In mitosis, the genetic material is divided equally, or the number of genetic material present in parent cell and daughter cell is equal. Therefore, it is known as equational division. While in meiosis, the number of chromosome or number of genetic material in the parent and the progeny cell is different. It is unequal. It is reduced. Therefore, it is known to be reductional division. So, coming to mitosis first. Mitosis, as said earlier, it is a equational division where the genetic material, there is an exact copy of chromosome in parent cell and two daughter cells. Mitosis is the process that partitions newly replicated chromosomes equally into two daughter cells. So this mitosis was the term derived from the Greek word meaning thread, and it was introduced by Walter Fleming. During mitosis, there is one round of DNA replication, one round of chromosome segregation, there is one round, and it generates two genetically identical daughter cells. So these are the criteria, or these are the characteristics. Mitosis is derived from the Greek word that means to be thread, and it was introduced by Walter Fleming. During mitosis, there is one round of DNA replication, one round of chromosome segregation, two genetically identical daughter cells are resulted. So stages of M phase. So we discussed G1 phase, S phase, G2 phase, and at last we have M phase. Right. In mitosis, the M stands for mitosis. If it is in case of meiosis, it would be meiosis phase. So now we will be discussing about the stages of M phase. Stages of M phase, particularly in which type of division? That is mitosis. This M phase starts with nuclear division. Nuclear division is termed as karyokinesis. So the stage of M phase, the M phase starts with the nuclear division, and next it leads to formation of nuclei, or we can say particularly formation of daughter nuclei. After the nucleus divides, it forms a daughter nuclei, and at last there is division of cytoplasm. I'll write it as cytoplasm division. It is also termed as cytokinesis. So these are the major events happening in the stages of M phase. So it starts from nuclear division, then it corresponds to formation of daughter nuclei, and usually ends with division of cytoplasm. Coming to mitosis context. Mitosis is a continuous process, and the M phase of mitosis is conventionally divided into 4 sub-stages, which are very much important. That is prophase, metaphase, anaphase and telophase. The mnemonics to remember this is PMAT. PMAT. So mitosis is a continuous process. They are conventionally divided into 4 substages: prophase, metaphase, anaphase and telophase. All these phases are derived from the Greek word. And pro means before, meta means mid, ana means back, telo means end. So these are the substages of mitosis, M phase of mitosis. So in our next class, we'll be discussing each phase, that is prophase, metaphase, anaphase, telophase in detail in our next class. Before ending this session, let's see the notes part. Mechanics of cell division. In eukaryotes, 2 types of cell divisions can be seen. It involves partition of genetic material into progeny of daughter cells. Mitosis is where parent cell divides into 2 daughter cells, okay, and each receives an exact copy of chromosome, genetic material in the parent cell. Therefore, it is also called as equational division since the number of chromosomes in parent and progeny cells is the same. Meiosis, the cell division where the genetic material must be precisely half so that fertilization will restore the diploid complement. So it is also known as reductional division, where it reduces the number of chromosome to half in the diploid number in the each daughter cell. Mitosis is a process that partitions newly replicated chromosomes equally into 2 daughter cells. Mitosis is derived from the Greek word meaning thread. It is introduced by Walter Fleming in 1882. 1 round of DNA replication followed by single round of chromosome segregation and generating 2 genetically identical daughter cells. So these are the stages of M phase. M phase starts with the nuclear division, karyokinesis, followed by formation of daughter nuclei, ends with division of cytoplasm, cytokinesis. There are 4 substage: prophase, metaphase, anaphase, telophase. So pro means before, meta means mid, ana means back, telo means end. In our next class, we will be discussing about each phase in detail. Thank you.